Hi everybody. So now we shall discuss some of the questions from genetic code. Right? Genetic code is a topic in molecular basis of inheritance. So let us start directly enter into the questions. The first question. Choose the incorrect statement. Rule of nitrogen base complementarity is followed during replication of DNA. It is true. It is correct statement. Right? RNA is formed on template DNA by following the rule of nitrogen-base complementarity. This is also correct statement. DNA is formed on RNA template by following the rule of nitrogen-base complementarity. This is also true. So 1, 2, 3 are correct statements. So naturally the fourth one must be the incorrect statement. Translation follows nitrogen-based complementarity between the sequence of nucleotides and the sequence of amino acids of a polypeptide. No, it doesn't follow the nitrogen-based complementarity, right? So, a particular nitrogen base cannot indicate a particular amino acid, right? It doesn't follow. This is the incorrect statement. So, fourth one must be the incorrect one. Let us see the answer. Okay. Rule of nitrogen-based complementarity doesn't exist between the sequence of nucleotides of nucleic acid and the sequence of amino acids of a polypeptide. Nitrogen-based complementarity rule is followed during DNA replication, RNA replication, transcription, and reverse transcription. Only during the formation of new nucleic acid and template nucleic acid strand only nitrogen-based complementarity is followed. In other cases, it is not followed. Coming to the next question. Is the matching. A code should constitute a combination of three bases. So this statement was given by a physicist, George Gamow. A4. It must be 4. Method for synthesis of RNA with a defined combination of bases. So, a chemical method was devised by Corona to form RNA with a defined combination of bases with homopolymers as well as copolymers. So, B3. Developed cell-free systems for protein synthesis. So, Nirenberg and his co-workers developed cell-free systems for protein synthesis. Right, so C is 2. The polynucleated phosphorylase. So this enzyme was discovered by Severo Oka, also known as Oka enzyme or Severo Oka enzyme. So D1. So A4, B3, C2, D1. Second option must be the correct one. Let us see the answer. Second one. Severo Oka discovered polynucleated phosphorylase, Oka enzyme. M. Nirenberg developed cell-free protein synthesis systems. Haragovind Khorana, chemical method for RNA synthesis with defined sequences. George Gamow proposed that a code should possess a combination of three bases. Third question. To which amino acid degeneracy of genetic code is not applicable? So, degeneracy of genetic code is not applicable to those amino acids which have only one codon to indicate them. If two or more than two codons are present to an amino acid, then it is degeneracy of genetic code. So let us see the which amino acid is given here. So tyrosine. Tyrosine has got two codons. So to tyrosine, degeneration of genetic code is applicable. Threonine. Threonine has got four codons. To threonine also degeneracy of genetic code is applicable. Tryptophan, yes, for tryptophan degeneracy of genetic code is not applicable because tryptophan has got only one codon, that is UGG. Even for methionine, the degeneracy of genetic code is not applicable. Methionine codon is AUG. So third option must be the correct one. Glutamic acid. Glutamic acid has got two codons. So tyrosine has got two codons, threonine has got four codons, glutamic acid has got two codons, tryptophan has got only one codon. So answer must be the 3. The 3 is given. More than one codons indicating the same amino acid is called degeneracy of genetic code, or redundancy of genetic code can also be called as. Methionine, AUG codon and tryptophan, UGG codon to it, have only one codon, and for them degeneracy of genetic code is not applicable. Threonine has four codons. They are ACU, ACC, ACA, and ACG. Tyrosine has got two codons, UAU and UAC. Glutamic acid has got two codons, GAA and GAG. Coming to the next question. Statements are given here. Given below are two statements. Statement 1, AUG codon has dual functions. Absolutely, it has got dual functions. It can be a start signal or initiating codon, and can also occur as interstitial codon, or the codon that occurs between initiating codon and terminating codons. So it has got dual functions, absolutely true. Codon of methionine can act as initiating codon. Codon for methionine is AUG only. Can act as initiating codon as well as interstitial codon, codon that comes in the middle. So statement 2 is also correct. Both statement 1 and statement 2 are correct. Fourth option must be the correct one. Let us see the answer, fourth one. Codon AUG is initiating codon, and it can also occur between first and last codons of the translated region of messenger RNA. It codes for the amino acid methionine. Methionine is a sulfur containing amino acid. Coming to the next question. Choose the incorrect combination. You have to choose the incorrect combination. UUG. So UUG is the codon for leucine. This is the correct combination. Leucine also has another one, UUA is also a codon for leucine. CUG. So CUG is also the codon for leucine. CUG is also codon for C. Leucine also has got CUU, CUC, CUA, CUG codons. So total 6 codons are there for leucine. UUC. So UUC is the codon for phenylalanine, not for leucine. So third must be the incorrect combination. Again, CUA for leucine only. Third one must be the answer. UUC is the codon for phenylalanine, but not leucine. UUA, UUG, CUU, CUC, CUA, and CUG are the 6 codons for the amino acid leucine. So other amino acids that have got 6 codons are serine and arginine. So only 3 amino acids have got 6 codons each. They are leucine, serine, and arginine. Coming to the next question. Assertion and reason. The codons of translated region of messenger RNA are read contiguously. Means in the coding region of messenger RNA, the codons are read one after the other continuously without any stop gaps. So this is correct only. Every nucleotide of coding region of messenger RNA is a part of one or the other codon. Absolutely true. So no nucleotide in the coding region is wasted. Every nucleotide is a part of one codon or the other. So assertion is correct. Reason is also correct. Reason clearly explaining why the codons of the translated region of messenger RNA are read contiguously, or genetic code is continuous or commaless. So A and R are correct, R clearly explained. So the correct option must be the second one. Both A and R are correct, and R is the correct explanation of A. Second one is given. Every nucleotide of the translated region of messenger RNA is a part of one or the other codon, and there are no stop gaps or punctuation marks. Going to the next question. If the sequence of nucleotides in a segment of translated region of messenger RNA is right. So this type of information is also given in the book. AUA, ACA, AAA, and AGA. So AUA, AUA, AUC, and AUU are the 3 codons for isoleucine. Should be isoleucine. First one must be isoleucine. Right? Next one, ACA. ACA is for threonine. ACU, right? ACC, ACA, ACG. These are for 309. 309 has got 4 codons. Right? Whereas isoleucine has got 3 codons. Next one. AAA. AAA is for lysine. Lysine has got 2 codons. AAA and AAG. Next one. AGA. AGA is one of the 6 codons for arginine. It also has got AGG codon. Right? AGG codon. It also has got CGU codons, CGC codon, CGA and CGG also arginine. 6 codons arginine has got. So AUA isoleucine, ACA threonine, AAA lysine, AGA arginine. All option must be the correct one. Let us see the answer. Third one. AUA, AUU, AUC are the codons for isoleucine. ACA, ACG, ACC, ACU are the codons for threonine. AAA and AAG are the codons for lysine. Lysine is a basic amino acid. AGA, AGG, CGU, CGC, CGA, right, and CGG are the codons for arginine. Coming to the next question. Identify the correct statements. Identify the correct statements. Codons that do not indicate any amino acids lack cytosine are cited in nucleotides. Codons that do not indicate any amino acids means they are nonsense codons. These nonsense codons lack cytosine because nonsense codons are UAA, UAG, UGA. So none of them is having the cytosine or cited in nucleotide. So A statement is correct. All nonsense codons have U or uracil or uridyl nucleotides. Absolutely true. All nonsense codons have got U. Right? U is the first one, first nucleotide in the nonsense codons. UAA, UAG, UGA. Right? So all the nonsense codons have got uracil and adenine. Right? None of them has got cytosine. 2 of them have got guanine. So second option is also correct. B is also correct. Chain initiating codon lacks cytosine. Absolutely true. Chain initiating codon is AUG. AUG is also not having cytosine. That means nonsense codons and initiating codons, both signal codons, start signals, stop signal, they do not have cytosine. That you have to remember carefully. Only one amino acid is indicated by 3 codons. Yes, only one amino acid is indicated by 3 codons, that is isoleucine. Already earlier we have discussed about these codons. The isoleucine has got the codons AUU, AUC and AUA. The last codon of the codon dictionary indicates amino acid glycine. Absolutely true. The last codon in the codon dictionary is GGG. GGG indicates amino acid glycine. Glycine also has got 3 other codons. GGU, GGC, GGA. Besides GGG. Right? So A, B, C, D, E. All the 5 are correct statements. So A, B, C, D, E. So A, B, C, D. Fourth option must be the correct one. Let us see the answer. Fourth one. Codons that do not indicate any amino acids are nonsense codons. They are UAA, UAG and UGA and they do not have cytosine. All of them have uracil and adenine. AUG is the chain initiating codon and it lacks cytosine and indicates amino acid methionine. Isoleucine is the only amino acid with the 3 codons known as AUU, AUC, AUA. The last codon in the codon dictionary is GGG and it indicates amino acid glycine. It also has got 3 other codons GGU, GGC, GGA. Coming to the next question. For change in the reading frame to read the translated region of messenger RNA, there must be. So when there is change in the reading frame due to mutations, it may be deletion mutations, addition mutations. So deletion mutations, how many bases are nucleotides? Addition mutations, how many bases are nucleotides? The reading frame is changed when in the coding region when there is deletion or insertion of 3 or 6 nitrogen bases now. If the three are multiples of three nitrogen bases that deleted or inserted, they do not change the reading frame because genetic code is triplet code. That's why there is no change in reading frame. Deletion or insertion of one or two or four or five nitrogen bases. Yes, if one base is deleted or inserted, two bases are deleted or inserted, four bases are deleted or inserted, five bases are deleted or inserted, seven bases are deleted or inserted, eight bases are deleted or inserted. There is a change in the reading frame. The second one is the correct one. Deletion and insertion of 6 and 9 nitrogen bases. 6 and 9 is the multiple of 3. So there won't be any change in the reading frame. Deletion and insertion of 3 or 9 nitrogen bases. It is also multiple of 3. So there is no change in the reading frame. So second option must be the correct one. Let us see the answer. Second one. Deletion or insertion of 1 or 2 or 4 or 5 or 7 or 8 nitrogen bases. Even 11 and 12 also you can, 10 and 11 also you can be example. Nitrogen bases changes the reading frame of coding region of messenger RNA and thereby the amino acid sequence is altered. Absolutely true. Deletion or insertion of 3 or multiples of 3 nitrogen bases doesn't change the reading frame of messenger RNA, but it changes the number of amino acids incorporated into the protein. This is true. Coming to the next question. Sickle cell anemia is due to change in the single nitrogen base pair in the gene for beta globin of hemoglobin at the 6th codon of the corresponding region in messenger RNA. Absolutely true. The codon GAG for the amino acid X is changed to codon GUG for the amino acid Y. X and Y are respective. Sickle cell anemia is due to change in the amino acid glutamic acid to alanine at the 6th position of the beta chain. So GAG is the codon for amino acid glutamic acid. Glutamic acid also has got another codon, that is GAA. And it is changed to GUG. GUG is the codon for valine. Valine also has got three other codons: GUU, GUC, GUG, GUA. Four codons are there. So glutamic acid is changed to valine. Second option must be the correct one. Let us see the answer. Second one. In the 6th codon, GAG for the formation of amino acid glutamic acid of beta chain of hemoglobin is changed to GUG to indicate the amino acid valine. It results in change in biconcave shape of RBC to sickle shape. Right? X is glutamic acid and Y is valine. It's a case of point mutation. It is a case of point mutation. It is a substitution mutation. It is. Right? It is transversion mutation. Right here in the DNA, the AT is changed to reverse. AT is changed to TA. This change is an example for base substitution point mutations. The transversion mutation as AT is changed to TA. AT is changed to TA in a corresponding region in DNA. One single nitrogen base pair substitution it is. It is a transversion mutation. Transversion mutation is a substitution mutation. Substitution mutations are a type of point mutations. Even single nitrogen base pair deletion or addition is also a point mutation. There are addition mutations also come under frame shift mutation. Point mutation, frame shift mutation, deletion mutation, addition mutation. Even substitution mutations are also point mutation. They are transversion and transition mutations. So please like, comment, share and subscribe. Thank you.